everyone. Let's begin. So yesterday in the first class, I tried to give a perspective, maybe a kind of slightly personal perspective of neuroscience, of computational neuroscience or theoretical neuroscience. I wanted to emphasize that theory in neuroscience is not like the kind of theories that you might have encountered in other subjects like, I don't know, mechanical engineering or electrical engineering, where theory is quite solid. The fundamental laws are pretty well understood and then they're mathematically formulated, and you know how to securely apply them to real world problems. In neuroscience, theory is there, vast amount of theory is there, but it's not yet of the quality of theory that you find in other fields. So that's a major challenge, and we try to convey as much as possible in this course about what is out there in terms of theory and computational tools in neuroscience. So before we take the next step, I just want to give a quick review of history of neuroscience, because one way to learn a subject is just look at all the basic concepts and the relationships and terminology and all that, which can be somewhat boring. But if you get introduced to the basic concepts through a historical account, I always find it's more interesting. So let us look at history of neuroscience very briefly, and this talk is titled "The Whole and Its Parts," because a major debate in neuroscience that has raged for a very long time is this question that is the brain function localized to different parts of the brain, or is it globally conducted and organized? And to this day, we are still a bit confused. We haven't found a definitive resolution of that debate even today. So with that in the background, let us begin. So perhaps you have a very skilled hand with which you can work wonders of color and form on a canvas, or you could be having great athletic abilities with strong legs that can propel you onwards at a whopping speed of 28 miles per hour, or you could be a model or an actress, right, with a face that can launch 1,000 ships, to borrow an expression from the English poet Christopher Marlowe. But thing is, we have all these qualities, might have all these qualities, but we often forget that this singular organ behind all these abilities of ours, that is the brain. That's because we can feel other organs, but we cannot feel the brain. It's silent. It's very active. It's active even when you're sleeping. Even the deep sleep, it is very active. It takes a big part, about 20% of the energy that is produced by the heart, or the blood flow, right, is taken up by the brain. So it's very active, but it's silent. Therefore, you don't give it the due credit, because it's not visible outside. But it is there, obviously, even if you're not sure about it, or somebody else is not sure about it. If you take a scan, that's what the brain looks like. So it's caged very securely, housed very securely in this bony cage, and that's been known for a very long time. And like I was saying, if you have a kind of a queasy stomach, it will make its presence known to you, right? We're producing all sorts of gurgling sounds. All right, or like Donald Duck here, if you're having thoughts of love, then your heart starts pounding and will make its presence felt. But brain is a silent organ, right? It's the controller of your personal universe, but it's a silent organ. So people have been interested in brain and understanding brain for a very long time, and have performed in the early days, performed some really crude experiments to understand what brain is like. So let us begin from the very early ages of humanity and see how they have dealt with brain. What were their attitudes towards brain? Let's start with ancient Egypt. So there was this papyrus found. They're supposed to be dated from 700 BC, or it could even be as old as 3000 BC. It's called the Edwin Smith papyrus, I guess, after the person who has discovered it. It talks about brain anatomy and talks about the meninges, which are like thin sheets of that cover the brain surface. It talks about ventricles. These are big cavities inside the brain. Some of the basic anatomical features of the brain were described in that papyrus. So Egyptians were very religiously or spiritually oriented people. I mean, according to at least the accounts that we have, and they used to believe in immortality, literally physical immortality. That is, so that's why they would mummify the bodies of the royal, members of the royal family. when they mummified, they would, so they think that heart is the most sacred organ, most important organ. That's the seat of the soul. This idea that heart is the seat of the soul persisted for a very long time, for millennia in history, right? Whereas for them, brain is not that important. So they would actually, they won't cut the head because that will violate the body, the integrity of the body. So they would scoop out the brain by inserting needles through the nostrils. You can see, as you can see in this picture. Then in ancient Greece, again, so let us start with Aristotle. Aristotle is one of the first physical philosophers. You know, there's a series of three great, Greek thinkers and philosophers: Socrates, Plato, and Aristotle. Socrates is the guru of Plato. Plato is the guru of Aristotle. Aristotle is the guru of Alexander, the Emperor Alexander. So Aristotle wrote extensively about a lot of physical phenomena. Right, he wrote volumes and volumes simply by observation and speculation. They didn't have the experimental method in those days. They would simply observe, apply logic, and come up with some theory of how it works. So for him, heart is the seat of the soul. That's the most important organ, right? So thing is, I mean, it has some meaning. To you can imagine why people would imagine, would assume that, because if heart stops, everything stops. I mean, the soul, body is dead. But if brain stops, right, somebody who's brain dead can still live on if you can, keep the lungs going and keep the heart pounding and so on. A person can still live on. So maybe that's the reason why he thought that heart is more crucial. But the point is, Aristotle speculated about many things in nature, and a lot of his theories were wrong. The real credit we can give to him is that the range of his thinking. He has covered a wide range of phenomena. For example, he has commented that when heavier objects fall faster than lighter objects, which is obviously wrong, right? I mean, unless you bring in the air resistance as a factor, right? The feather falls slowly than a stone, which is fine, but that's because of air resistance. But if you remove that, both feather and stone will fall at the same speed. But anyway, so his speculation was wrong, and much later Galileo has disproved him by a classic experiment that he did from the Tower of Pisa. Then in second century, again, Greek physician called Galen, that has written a book called On the Brain, in which he described the brains of a wide variety of animals, including cats and dogs and weasels, camels, lions, wolves, bears. So apparently he has spared insects. The insects do have brains, they're very small, and to work out their nervous system, you need microscopic devices. All right, so obviously Galen didn't have that. So I guess he's limited himself to the study of slightly these larger animals. So in this book, he talks about how to perform dissection of, for example, here he's talking about dissection of an ox's brain. Right, he says when the brain is suitably prepared, you see the dura mater is one of the meninges, right, the surface of the supporting this coats of the brain. Then slice straight cuts on both sides of the midline down to the ventricles. You will observe a third ventricle between the two anterior ventricles with a fourth behind it. So it's like a proper anatomical text, right? But the problem is, so Galen had done extensive surgeries or dissections on cadavers, animal cadavers, but he later extended them to even human brain, which is wrong, right? I mean, although mammals share certain features, common features, you cannot just blindly extend the knowledge of an ox's brain to human brain. So that flaw was discovered later by Vesalius. So it's a long story. So later on in the Islamic world, in the the later half of the first millennium, 700 to 1000 AD and all. So some scholars have kept the lamp of knowledge glowing. So for example, there's Al-Zahrawi, who used surgical tools to perform lots of brain surgeries. So for example, head injury people or brain fractures, right? He would use lots of different instruments. He described how to cleanse them, how to maintain them, how to use them, and so on. So if you fast forward a little to come to Renaissance period, that's about 500 years ago. So thing is, since about Aristotle's time, a little bit after that, maybe after the fall of Roman Empire in the middle of the first millennium, up to the middle of the second millennium, that period is considered to be the European Dark Ages. That was considered to be the medieval period, where so it all depends on the kind of beliefs, basic beliefs about life that society had at that time. So religion had a very strong kind of hold on the minds of people in that period. That's why it is called medieval period. That was one of the reasons. So because of that religious thinking, there was a certain philosophy of life, which went something like this. The teaching is that the purpose of life is to do good deeds and reach heaven at the end of life. Life itself doesn't have much meaning or intrinsic purpose. Its whole purpose is that it will give you a chance to perform good deeds, and so that you can go to heaven after life. So it's like you sit in a waiting room for the train to come, right? You don't give much importance to the waiting room, right? The interior decoration and facilities and all that, because it's only a temporary sojourn that you are spending in the waiting room. So world is like that, right? When you have this kind of religious thinking, world is a big waiting room. Life is big, just a waiting room where you wait, sit for a few hours before you get your train and hop onto the train and head out. So when philosophy of life is like that, you won't have much interest in life or world or nature. You won't try to understand, right, what all is there in the waiting room. So that kind of introspection began in the Renaissance. It slowly began from about 12th, 13th centuries, and after that, a lot of people kind of started sharing those ideas, and then they said, why are we thinking about world like this? Right? Why do we never pay attention to the world and nature? Why don't we study nature? Why do we blindly believe these ideas about afterlife and so on? So the lot of translated work from the Arab world and classical Greek and Roman literature, right, also has fueled this new thinking, and so that led to a huge amount of creativity in art and music and architecture, sculpture, and so on and so forth. Like, it's pretty much an outburst of creativity in pretty much all human endeavors, right? So in that process, people also have started having new thoughts about brain and mind and things like that. A key figure in Renaissance is Leonardo da Vinci, Italian from Tuscany. So Leonardo, we know as an artist, the artist of Mona Lisa painting, but he is also a scientist, an engineer, and he's done extensive work in optics and mechanics and fluid flow, and also done a lot of engineering work. He's also done a lot of studies on anatomy, human anatomy. Because why does an artist have to study anatomy? Because he has to understand the human form very accurately, so as to reproduce it in his artwork. So he would actually buy the cadavers of executed criminals and kind of smuggle them to his studio stealthily in the night, and then he would dissect them and study the internal organs. So through such experiments, he has done extensive work on anatomy, and he's also done a lot of work on brain. So he knew about brain surface and its convolutions and the ventricles, right, the nerves which lead out of the brain to various parts of the body, and so on and so forth. Then came Descartes. So Leonardo da Vinci also had extensive understanding of anatomy. It is gross anatomy, not microanatomy. But when it comes to when it comes to function of the brain, everybody thought that there is some kind of energy, some kind of animal spirit that makes the brain active, and that's what controls the body. So this kind of a strange idea about some kind of animal spirit was there for a very long time. So people had to insert the idea of God, randomly everywhere, wherever they're not sure of an explanation, they would randomly insert God. So this insertion of God became a big problem, and for a long time, and it kind of inhibited the development of science, because it's been there. It's very strong during the medieval period, and Galileo, who was the father of modern science, had to struggle with it all his life. He really paid through the nose for his constant conflicts with the church, for example. So, for example, if you ask, like, why are the planets round? The explanation is the sphere is a perfect shape, and God likes spheres, and therefore planets were created to be round, right? Why are planetary orbits circular? Because again, circle is first of all, they're not circular, but then they thought it's circular. So circle is a perfect shape, geometric shape, and God likes perfect shapes, and therefore, so like that, for everything they would randomly introduce a kind of invoke and give credit to God, right? So this kind of random insertion of God idea in every theory, right, became a problem that was inhibiting science. So somebody had to deal with it, and René Descartes, French philosopher, he has dealt with it in a very interesting fashion. He said, yes, we agree that God created the world, but once he has created the world, he has like, you know, just as a watchmaker, right, makes a machine, right, or a watch, and he keys it in. After that, it keeps ticking on its own. There's no need for a watchmaker to watch over it and intervene at every moment. So similarly, once the world is made like a machine, it will keep ticking, following certain laws, fixed laws. God doesn't interfere in the operations of the world. So that kind of made the two parties happy. Church is happy because, you know, the ultimate credit is given to God. People with more scientific bent of mind are also happy because now they don't have to worry about God. They only have to worry about the laws of nature. So that kind of triggered or kicked off the scientific revolution, and a lot of progress also was made in allied fields of neuroscience. So there are certain fields which contributed to neuroscience, and developments in these fields occurred very rapidly since that kind of a intellectual breakthrough was made. So one of the first lines of work that we can mention is the microscopic observations of brain cells, and so Robert Hooke made his first observations of the cell in a microscope. You know, you could take this onion cell, you know, onion skin and cork and all that, and found that the living matter is made up of special distinct units and called cells, right? And they had a wall around them and so on. So which was a very major discovery, right? So people also began to make up microscopic observations of brain tissue, right? And but thing is, very early on, people felt that the brain tissue is slightly different from other tissue, because other tissue always had some kind of a cellular structure. They're divided into small modules. But brain tissue does seems to be slightly different. There doesn't seem to be any differentiating wall from one side to another. They all seem to be connected, right, continuously through, like, you know, through some kind of a corridor going from cell to cell. It's kind of like your HSB building, for example. In HSB, you have at one extreme you have the math department, you know, the PRO's office and all that stuff, the placements office and all that. Then in the middle you have the humanities department. In the other extreme you have the physics department. But they're all connected by corridor, right? By corridors, right? So they're not distinct entities. The people thought that the brain cells or neurons or something like that. You know, the neuron name came a bit later, but so people thought that they're all connected by some kind of corridor. The reason they felt like that was the earlier microscopic observations used to look at the raw tissue, right? So other tissues have some color. For example, skin has certain color. The red blood cells have red, you know, but whereas brain cells, right, are almost colorless. They're almost transparent. So it's very hard to make out the differences or the differentiating boundaries of neurons. So that ability came with this interesting staining technique developed by Camillo Golgi, right? And with this, we developed what is called now the Golgi stain, which is now used extensively in biology labs all over the world. So Golgi has also stained the hippocampus, which is one of the important brain areas involved in memory. And so you could see this small. So you see that the stained parts are actually cells; they are neurons. You could see them quite distinctly. So that was a major breakthrough. So they were not able to see neurons so clearly before the advent of the staining technique. So Ramon y Cajal, right, Spaniard, right, he has taken Golgi's staining technique and applied this to lots of different brain tissues and studied the cells. And his conclusion was that brain tissue is not a syncytium. Syncytium is a bunch of cells which form a continuous system, like a corridor, right, which have a corridor like continuous connection. So he concluded that brain tissue is not a syncytium, and brain cells are distinct like any other cells that we have seen before, and they are individual entities, and he called them the neurons. So on the right, you see these are Purkinje cells from cerebellum, a kind of neurons found in cerebellum. The thing is, these are hand-drawn pictures. There's a beautiful artwork. I mean, even now, Ramon y Cajal's pictures, hand-drawn pictures of neurons are like used as masterpiece of art because they're very beautiful pictures. And now we don't draw pictures; we just do micrographs. We just take pictures, photographs from using a microscope. But the thing is, although Ramon y Cajal kind of concluded that neurons are distinct entities and there is a clear border separating one neuron from the other. In fact, that idea is called the neuron doctrine. Right? Thing is, he didn't have complete evidence to say that at that time, because unlike other cells, so if you look at red blood cells, they don't necessarily come very close to each other. They don't stick to each other for a very long time, because if they stick to each other for a very long time, that's a blood clot and that's dangerous. So they maintain some distance. So you can see very clearly that sorry, how one cell is distinct from the other, how separate from the other. But in case of neurons, they make these deliberate contacts with each other, and where these contact points are come very close to each other. This is a problem. This was a major difficulty, right, in understanding that neurons are actually distinct entities because they come very close to each other. At this point of contacts, which we now know as synapses, the distance between the two neurons can be as small as 20 nanometers. Okay, so that's a very small gap to be discerned in the days of Ramon y Cajal. He didn't have that kind of microscope. You need to have electron microscopes to see such small gaps, as you can see in this microscope picture or a microgram. So like that, at the level of, so for millennia we had knowledge of brain's gross anatomy, but only in the last couple of centuries with the improvement developments in microscopy and the staining technique, actually last century, we started understanding the microstructure of the brain at the level of microscopic structure. What does the brain look like? What is it made up of? Then when it comes to function, we need to go back to the earliest experiments of Luigi Galvani. So Galvani was studying electrostatics. This was the days when we didn't know about electric currents. Ampere came much later. So he was, how to produce a charge by, rubbing certain materials and all that, right? You studied about that in your high school. So he was taking these charge rods, and once he was applying it to a frog's leg. So they've taken a dead frog, kind of skinned it, peeled off the skin, and exposed the muzzle. So when he applied this charge rod to the muzzle, the frog's leg twitched as if it's still alive. It's a dead frog, obviously. It means only half the frog is present. He was quite shocked because, literally, no pun intended, because how could a dead frog move like that? And only the charge was making it move. So then he understood some connection between electricity and life. He thought that it's electricity that's making life possible, right? Making us move and so on. So now that idea of electricity and connection with electricity and brain's activity or muscle activity, that became clear. And so the connection between electricity and brain activity also became clear later on. And so that was very nicely captured by the development of this device called the electroencephalogram. This was developed by Hans Berger. So he has placed electrodes on the scalp of the brain, the scalp of the head, right? And these electrodes can pick up brain's activity and what is popularly called brain waves, right? And so, so also the electrical nature of the brain was known like that for a long time, throughout the first half of last century. But exactly how is this electricity produced by the brain tissue? How do neurons produce this electricity? So that was not known for a very long time. So British physiologists Alan Hodgkin and Andrew Huxley has taken up this question, and they wanted to study how a neuron produces electricity. What is the molecular machinery, right, or cellular machinery that can produce electricity in brain tissue? So for that, they need to actually take recordings from a neuron, right, to understand how it produces electricity. But normal neuron, let's say human neuron, is very small. And if you want to study the conduction of the signal, electrical signal, along one of its fibers, the fibers are even smaller, right? A few microns thick. So Hodgkin didn't have that kind of a microscopic electrodes needed to penetrate such small volumes. So they wanted a big neuron. So where do you get a big neuron with a thick fiber, right? So they got hold of the giant squid. You might have seen the giant squid in the Pirates of Caribbean, right, movie series, right? You see this huge animal, you know, sinking ships and all that. So they have taken one such giant squid and they brought an axon segment and they performed experiments on it. And they found that axon has a very peculiar electrical response properties, right? If you inject current into it, and if you it has, first of all, it has a voltage always between inside and outside. And if you inject current into it, it produces a kind of a spasmodic response, an outburst of response, where by which the voltage suddenly goes up to some peak value and comes down. And goes back to the baseline voltage. So this sharp spike of a voltage is called the action. They called it the action potential. They also worked out, so what kind of molecular machinery is present, right, and what kind of chemicals are. The chemicals play a major role in the production of this kind of this action potential. So we'll study all that later. So like that, there's a lot of progress in understanding how electrical signals play a key role in the function of a neuron. But what was not clear for a very, very long time, right? How do neurons talk to each other? How does a signal go from one neuron to another? Because if neurons are distinct entities, there is actually a gap separating one neuron from the other. What processes that gap? Is it some kind of electrical charges that go from one side to another? Does one neuron release electrical charges which will activate and produce electricity in the other neuron? Or is it a chemical? So this was a very hot question because you could not just poke that gap with an electrode or something or a sensor and measure what is out there, because it's a very small gap, just 20 nanometers. So the hypothesis that a neuron could be releasing a chemical, right, and to activate produce an action on another neuron uh, had its basis in other experiments or other data that we have historically. Because even for a very long time, the hunters used to use poison arrows, right, and to immobilize or kill an animal. So these arrows are tipped with a certain chemical called muscarine. So this chemical would act on when the arrow gets rooted in the muzzle of the animal. This chemical would act on the muzzle and immobilize the muzzle. So these chemicals are actually neurotoxins. They kill neurons. They also deactivate muscular action. So that's a simple example of our demonstration of how some chemical can act on muscle, right? And so the chemical action on the brain also is known in an indirect fashion. For example, if you take alcohol, like you have a glass of beer or something, you feel lightheaded and so on. So which is simple proof that chemical can act on your brain and change brain function. So those kinds of generic data is there pointing to a fact of a chemical action, but they didn't have any concrete proof. So Paul Ehrlich has suggested that when a chemical acts on a cell, like when neuron to some other cell, right, it may not be directly, so it may be having some kind of a specificity. That is, certain chemicals can act only on certain cells. There's some kind of a preferentiality in the action of a chemical on a cell. Right, and if you understand that well, that can be a basis for a lot of cures, because very often the disease is caused by certain microorganisms. They're also living cells, right? So if you know what kind of chemicals can act on those such living organisms, those microscopic living organisms, then we can come up with the toxins which can ride over these chemicals, and along with the chemical they'll go and get themselves attached to the microorganism and kill it. Right. So that's a very nice idea, and he called this idea the magic bullet. Then John Langley thought of proposed the concept of a receptor substance. Basically, what he is saying is when a chemical acts on a cell, right, it doesn't act directly on the cell, but it acts on another molecule which is present on the cell. Okay, so this molecule which is present on the cell, he calls it a receptor or receptor substance. And the other chemical which is acting on the cell, these two have some kind of a specific affinity. That is, only a certain chemical can act on certain receptor. Right. So this is how you get special affinity or specificity, and this can be used to design drug molecules. Again, the kind of things that Paul Ehrlich was talking about, the magic bullet. The drug is a magic bullet which can specifically act on a cell through the agency of a receptor. So all these ideas were around, but a definitive proof or a demonstration that the action of one neuron on the other, right, is through chemicals, was missing, or the action of a nerve on a target organ. So that one of the first experiments of that kind, a really brilliant experiment that was done by Otto Levy. I think he's an Austrian physicist. So apparently he was thinking about this problem of demonstrating the chemical action of a neuron, and for a long time. And one day he was working in the lab and he slept off in the lab, and that apparently is how the story goes. He had a dream in that night and about an experimental setup which can very nicely demonstrate this principle. So he kind of got up in the morning, but he didn't remember the details of the experiment very clearly. So he felt very bad for not getting up at that time and noting down the experiment. So next night, apparently he got the same dream again. And this time he had the good sense to wake up and note down the experiment. Right. So next day, following day, when he woke up, he remembered because he noted down what he dreamt of. He put together the same equipment and performed the experiment. He got brilliant result. What was the experiment? It's very simple. He took a frog heart, right, which is placed in a ringer solution so that it can beat spontaneously, and this can be done. So now the heart is prepared so that it will have the natural nerve connection. So heart is controlled by a nerve called vagus nerve, right? And if you activate that nerve, heart slows down, the beat slows down, so which is well known. So he's taken out the heart of a frog along with the vagus nerve intact, and preserved it in ringer solution so it can keep on beating. So now he electrically activated the vagus nerve, and then the heart naturally slows down. So then he has taken a little bit of the solution from the first beaker, right? And in the second beaker, he had another heart which didn't have the vagus nerve. He just transferred some liquid from the first heart to the second heart. Now the second heart started slowing down, which is very interesting. But how do you explain this result? Because when the nerve is activated, right, the heart slowed down. But in second case, there is no nerve. There's only transfer of fluid from first heart to second heart. Yet the heart slowed down. How did this happen? The only way to explain this is when the nerve was activated, the nerve released some kind of a chemical, right, which acted on the first heart, but also a bit of it leaked out into the surrounding medium. And therefore, when the fluid was transferred from the first beaker to the second beaker, right, it was able to act on the second heart and slow it down. So which means the agent that mediates between the nerve and the organ, the heart, is a chemical. So which was a brilliant proof. And so this is one of those classic experiments where you demonstrate something really fundamental, really profound, using an experiment which is quite trivial. I mean, look at how much does it cost to perform the experiment? Maybe a couple of $100, that's all. Right, and all the drama that we make these days about not having enough funding, and we need crores and crores and crores to find something very simple. Okay, so that gave us the idea of a chemical synapse. That is a synapse which is a junction point between one neuron and another. Right, uses a chemical signal to talk to the other neuron. So that's a chemical synapse. But people also have subsequently found electrical synapses, where two neurons actually literally join their membranes, right, very tightly, right, and actual currents, ionic currents, flow from one neuron to another. This is kind of like sensation. It's not so crude like the original conception of sensation, because these currents flow through very fine channels formed by proteins, right, or what are called gap junctions. But anyway, it's something like the sensation. So like that, when it comes to microscopic structure and microscopic function, a lot of things were known. But what about gross function? How does the brain work? Right, let us look at the history of that. And earliest developments in that area came through in the form of this kind of a very crude and pseudoscientific body of knowledge called phrenology. So phrenos is head or mind, and logy is study of mind. Study, right? So it's the study of mind. So this was proposed by a guy called Franz Gall, right? I think he's an Austrian. So what these guys would do is they would their arguments go something like this. They would tell the character of a person by feeling around the shape of the head. So this is kind of like palmistry and in astrology, things like that. So their argument goes something like this. So everybody has certain characters, right? Some characters are stronger in some persons than others. Now, different characters have different bases in the brain, right? For example, let's say generosity is in your near the temples, right? Or your memory power is in your front of the brain. Some nonsense. I'm just picking up something. Now, if you have very strong memory, then that part of your brain is larger, more swollen than others. Or if you have very high generosity, that part of the brain which represents generosity is fatter, it's more swollen than in case of others. Okay, so now, okay, then we go to next step in the logic. So these differentially swollen areas of the brain keep pushing outwards, right, into the skull, into the cranial wall, and produce little bumps in the skull, which can be felt by feeling the surface of the head. So therefore, if a person has high generosity, I'll just feel around the generosity part of the skull, right, of the skull, and I'll know that this guy is more generous than others. This whole thing is totally rubbish, right? But this is the kind of, so they would even make these charts and kind of hoodwink a lot of people. Even top people in society would fall for them, and this guys used to make a lot of money. So thing is that people are okay with these people telling nonsense, but people are not okay with these people making a lot of money, right? That upsets some other people. So Napoleon didn't like this all nonsense, because Napoleon was not only a kind of avaricious emperor, who was expanded, his land and created large empire and all that. He's also a great votary of science. He wanted to create a strong scientific temperament in his empire. He didn't like the work of Franz Gall and all this phrenology nonsense. So he said, let us investigate this thoroughly, right? Let us see if anything that these guys are saying, is it scientifically valid at all? So he called one of the court scientists called Jean-Pierre Flourens and asked him to investigate this problem, scientifically. So what Flourens has done is he has taken experimental animals, right, and he had exposed their brains and damaged the brains in different parts, just to see if uh, damage in distinct locations causes distinct behavioral changes in the animal. So he did that and he couldn't find any distinct changes, and because what he observed was that no matter where the damage is, the animal showed the same level of impairment in its behavior. What really matters is the extent of the damage and not the location of the damage. That was his conclusion. So, but later on, British neurologist called Hugh Jackson, right, thought of the problem very differently. He was studying seizure patients, and seizure patients, very often the seizure starts from one extremity, like let's say the fingers of the hand, and the tremor slowly spreads next to the wrist, and then to the elbow, and then to the shoulder, and then to the whole body, and the person loses balance and falls down. So there's a peculiar pattern by which the seizure spreads. So he saw that, and he had a brainwave. That is, if the seizure spreads like this, then what could it tell us about how the brain controls the body? So he thought that maybe there is a part of the brain which controls movement, right, and also maybe nearby parts of the brain control nearby body parts. So, for example, the part of the brain which controls the hand, the fingers, is right next to the part of the brain that controls the wrist, which is next to the part of the brain that controls the elbow, and so on and so forth. So there's a kind of a continuous mapping from the brain to the body surface, so that nearby body parts are controlled by nearby brain regions, right? Why did he assume that? Because when the seizure starts, maybe some kind of abnormal activity starts from one part of the brain, right? Let's say from the finger region of the brain, and from there it spreads to neighboring regions of the brain. So it spreads to the elbow region, spreads to the wrist region, then elbow region, shoulder region, and so on. The way a fire spreads in a forest. So therefore, you see this kind of a systematic spreading of the seizure activity of the tremor, right, in the person's arm, in the hand and arm and body. which was a beautiful insight. He is quite right in that observations. And so therefore that approach, right, that thinking has led to the idea that brain function is localized, right? Different functions are performed by very distinct localized parts of the brain, right? So this was totally opposite to what Jean-Pierre Florence has concluded. So similar data has come from other studies. Pioneering among them was for the studies of Wilder Penfield, the Canadian neurosurgeon, where he found he was performing experiments on again epileptic patients. In these people, there is a abnormal activity that starts in one part of the brain, spreads all over the brain. So therefore, as a manifestation of that, the abnormal motor activity also starts from one part of the brain and spreads to all over the body. Sorry, starts from one part of the body and spreads all over the body. So one of the surgical interventions for this problem is to isolate the starting point of this activity called epileptic focus and surgically remove that part. So hopefully that will solve the problem, and that was working in many cases. So in that process, he would to identify this epileptic focus, he had to electrically stimulate different parts of the brain with an electrode, and then see stimulation of which part of the brain triggers epileptic seizure. Okay, so in the process, he was also studying, he was also stimulating different parts of the brain to see what happens to the patient. So when he stimulated certain part of the brain in the occipital lobe, in the back of the brain, the patients would report seeing flashes of light. Occasionally, when he stimulated certain part of the temporal lobe, the patients would report hearing sounds or sometimes even songs from long ago, right? Or if you stimulate the part of the brain, what's called the post-central gyrus, they would feel that somebody is touching them. So depending upon where the stimulation occurs, they would feel that somebody is touching them at various points in the body. So all this has revealed that different stimuli or experiences are encoded in different parts of the brain. So this was really path-breaking experiments. So later on, people were able to work out that there are indeed such kind of brain maps or maps of the body on brain surface. So you can see here that there's a continuous mapping, or nearly continuous mapping between, right? If you look at the left one, there is face and nose and eye and thumb, index finger, middle finger, ring finger, little finger. So it's kind of continuous mapping between the body surface and the brain surface. These maps do exist. So this was a big breakthrough in our understanding of brain function, and people got very excited about it. They started making these little figurines just to indicate or convey this idea, this major discovery of brain function. So they thought that there's a little kind of a man sitting inside the brain controlling our body. So they called this little man homunculus. Homunculus means little man. And they made idols of this little man, imaginary idols. So other studies by, for example, Paul Broca, French neurologist, also has revealed interesting data about how speech and language are organized in the brain. So Broca studied interesting class of patients. Right, one of these patients, he could only utter one word. That word is tan. He could only say tan. Maybe he would say tan. That's all. Right, and so after the death of this patient, he performed postmortem and found that in this patient there is a lesion or a damage to a specific part of the brain, which was later called the Broca's area. We'll study about all this. No, maybe not. Maybe if we have time, I'll describe this later in the course. So similarly, this other kind of a language dysfunction studied by Carl Wernicke, right? And these people, so the Broca's patients could utter single words but could not frame full sentences. Wernicke's patients have the opposite problem. They could utter full sentences, but they don't have much meaning. Right? For example, Wernicke's patient says, "I called my mother on the television. I did not understand the door. It was to breakfast, but they came from far too near. My mother is not too old for me to be young." So like that, there's no connection between one sentence and another. Even single sentences don't have much meaning. So this is the opposite problem. This is called the Wernicke's aphasia. And Wernicke's aphasic patients, and after their postmortem studies have revealed that they have lesions somewhere shown in the picture, the outline with this outlined by the white line. Okay, so there is a part of the brain which is responsible for language production, right? And the other part of the brain which is responsible for language understanding or comprehension. Then there's a third kind of aphasia found, right, where the people can understand what is said to them. They can also say things which are meaningful and form full sentences, but they cannot repeat what is said to them. Okay, it's a very peculiar kind of a language problem. Aphasia means a language problem. So in these cases, this patient, it was subsequently found that the bundle of fibers which connect the Wernicke's area to the Broca's area, there's a damage. So it's very clear. I mean, brain is like a very nice machine, like a large circuit, right? So different parts of the brain perform different functions, but sometimes they have to work together, right, to perform a larger function, and they work together through by conveying signals through these bundles of fibers, right, and so on. So that kind of a picture came with the studies of Wernicke. So Wernicke tried to put an end to this long-standing debate or a fight in neuroscience about two opposite views. One view said everything in the brain controls everything. That's called the aggregate field view. Other view said no, different parts of the brain perform different specific and exclusive functions. That's called the localization view. So Wernicke tried to put a stop to this debate, or want to give a resolution to this debate, and said that yes, different parts of the brain perform different functions, different simple and elementary functions. But so, for example, language understanding is done in Wernicke's area, right? Language production is done in Broca's area. But if you look at language function as a whole, that is done by both of these areas and their connecting fibers, and actually more and other structures in the brain. There's a whole system, right, of the brain which conducts your language operations. So like that, Wernicke has kind of proposed the idea of parallel and distributed processing. So he said that brain function is connected by parallel and distributed processing. So to perform any large scale function, many different areas in the brain should work together, work in parallel in a distributed fashion. So although these ideas were proposed way back in the middle of 19th century, right, to give it a mathematical form, we had to wait until the 80s, actually, right, very recently, just 4 decades ago. So in the 80s saw this, what is called the connectionist revolution. That is, they said that in the brain, the real players are not the neurons, but the connections between neurons. All the information is there in between the neurons in the form of connection, the synapses. Right? All the knowledge is encoded in the connections. And later on in the more recent times, maybe last half century, right? There's a lot of progress in technology also. Like people are able to record from single neurons, not only single neurons, but single ion channels or small number of ion channels. Like there's a technique called FastClam that was developed. And lots of different imaging techniques have been developed again in the last few decades. And so people are now taking up this ambitious projects like the Human Connectome project, right, where people are trying to work out the entire connectivity graph of the adult brain. People are also trying to build large-scale brain models. Right, there's this ambitious project, computational neuroscience project, called the Blue Brain project, which is done in this research center in Geneva. So here their goal is to build a computational model of the brain as it is, in all its detail. Right, every neuron, every fiber, and every channel, and so on and so forth. It's just a super ambitious project. They have started with smaller animals, smaller brains, but ultimately they want to reconstruct literally the whole human brain. So it's an exciting field, lots of open questions, and in practically infinite amount of data available. Although the problem is we don't fully understand the data. Right. So in the course, I'll try to focus on fundamental principles. I'll try to explain present models which are more crucial, which capture the fundamental principles, and I'll try to give some tools. And after that, it's up to you if you want to study a certain part of the brain, certain system. You can use these tools and study them. But my emphasis will be on system neuroscience and not so much on the single neuron modeling. Although I'll spend a lot of time on single neuron modeling, my emphasis will be on system neuroscience and some of the large scale questions, because I feel strongly that we need to make a lot of progress in our understanding of disease process, disease spreading, and all that, and how do symptoms develop. and use computational models to actually treat brain disorders. Right? So for that, you need certain kind of models if that's your objective. So you'll see that kind of a slant or a bias in the material that I'm presenting in the course. So so I think Friday's class, is like your workshop. So please attend the workshop and see if you can enjoy some of the talks. Then from next week, next Monday, we'll start with a bunch of mathematical preliminaries. We'll cover some basic ideas from dynamical systems, and then we'll move on to single neuron biology. Okay, so that's all from my side for today. And if you have any questions, I can take them. Hello, anybody?
Sir, how do neural links work?
Sorry,
Neural links.
Neuralink, the company.
Yeah, it's about attaching a chip to brain.
Yeah,
Bro.
yeah. I don't know much about Neuralink. I mean, I can maybe I'll try to talk about it towards the end of the course. All right. Hello.
Okay, sir. Thank
Yeah.
you.
Okay, bye. Anything else? Okay, then have a good day and see you
Yeah.
in the workshop.
Thank you.
Thank you. Bye.
Thank you, sir.